Hello, students! In today's class, we are going to understand an important molecule involved in protein synthesis, tRNA, or transfer RNA. But before we study its structure, let us understand a very interesting question. Why is tRNA called an adapter molecule? To understand this, we need to go back to the time when scientists were trying to decipher the genetic code. Francis Crick realized that there had to be a mechanism that could read the genetic code and connect it with the correct amino acid. Why was this necessary? Because amino acids themselves do not have the structural features required to directly recognize specific codons. Therefore, Crick proposed the existence of an adapter molecule. This molecule would perform two functions. First, it would recognize the genetic code, and second, it would bind to a specific amino acid. tRNA performs exactly this role. One end of the tRNA contains an anticodon, which has bases complementary to the codon on mRNA. The other end contains an amino acid acceptor site, where the specific amino acid is attached. So tRNA acts like a molecular bridge between the language of nucleotides and the language of amino acids. That is why tRNA is called the adapter molecule. Interestingly, tRNA was known even before the genetic code was proposed. At that time, it was called sRNA, or soluble RNA. Its role as an adapter molecule was understood later when scientists began to understand how the genetic code connects nucleotide sequences with amino acids. Now let us understand the structure of tRNA. The secondary structure of tRNA is commonly represented as a cloverleaf structure. The most important regions for us are the anticodon loop and the amino acid acceptor end. The anticodon loop contains three bases called the anticodon. These bases are complementary to the codon present on mRNA. At the opposite end is the amino acid acceptor end, where the specific amino acid attaches. Let us take a simple example. Suppose the mRNA contains the codon UUU. A tRNA with the complementary anticodon AAA can recognize this codon. This tRNA carries its specific amino acid, phenylalanine. Therefore, the tRNA recognizes the codon through its anticodon and delivers the correct amino acid to the growing polypeptide chain. This is the adapter function of tRNA. Now, remember these important points for your board and NEET examinations. First, tRNA is called the adapter molecule because it connects the codon with its corresponding amino acid. Second, the anticodon of tRNA recognizes the complementary codon on mRNA. Third, the acceptor end binds the specific amino acid. And one very important point, there are no tRNAs for stop codons. Although we commonly represent tRNA as a cloverleaf structure, this represents its secondary structure. In reality, the tRNA molecule folds into a compact three-dimensional structure that resembles an inverted L. So remember, cloverleaf, secondary structure. Inverted L, actual three-dimensional structure. So to summarize, tRNA is known as the adapter molecule because it connects the genetic information present in mRNA with the correct amino acid. Its anticodon recognizes the codon, while its acceptor end binds the specific amino acid. Its secondary structure appears like a cloverleaf, while its actual three-dimensional structure resembles an inverted L. And remember, there are no tRNAs for stop codons. In the next part, we can explore how tRNA participates in the actual process of translation and protein synthesis. Pause the video and go through these questions for better understanding of the topic. Thank you for watching this video. I hope you now have a clear understanding of tRNA, why it is called the adapter molecule, and its important structural features. Keep revising, keep practicing previous year questions, and keep strengthening your biology concepts. Thank you for watching. Keep learning, keep growing, and I'll see you in the next class.